I assume I did the I assume you do a 5 or midnight. Can I borrow a pen? I'm gonna write it on my hand. Wasn't governed yet.
I'm just gonna leave this up on my computer.
I'm glad you reminded me. Thank you.
I remembered as he handed this back, I was like, at first I thought he was almost gonna turn off yours.
So where's the cat? Where's your cat?
Cat.
There's the cat.
I love cats. My cat is finally not sick anymore, and I'm so happy.
Life is better with a kitty.
The quiz for this class is due at the end of the day, right?
Yeah. Yeah.
We were busy studying for PCAT. So.
Yeah, the quiz is just 5 questions. You should be able to do that without much of a hassle.
We like 10 minutes to take it.
10 minutes. Should not be a big deal.
Yeah. And the exam, just to double check, is just over lipid and cholesterol.
Yeah. Yeah. Which is more than enough.
Make sure I don't have more to study. Is it gonna be 50 questions again?
Yeah. Okay. All right. Hi, kitty. All righty. As soon as Jenna gets here, we'll.
She's right out of the bathroom. She's
Okay.
going to go to class.
I hope everyone got a chance to enjoy this last weekend. It was glorious. There's no two ways about it. It was not too hot, not too cold, not at all humid,
Yeah.
very slight breeze.
My dog has been really upset because he's staying with me and he can't play frisbee because I don't want to let him off the leash with there's beer with in the apartments. And so the track was empty on Sunday, so we can play frisbee on the track, and he was so happy.
Yeah, it was amazing. It was an amazing day.
That's the nice thing is my dog is forcing me to go outside and take walks even if I need to be
You know, in Great Britain, if people have a weight problem, one of the things the doctors will prescribe is get a dog because it forces you to walk every day. And so that habit of walking, if you don't have that habit, they'll just say, well, get a dog.
Yeah, I walk like a mile almost every day that we've been here.
Easily. Probably more than that.
Yeah, more.
And
than
I'm
that.
sure for every mile you walk, he does too. All righty. Let's get started. Dane, you feeling any better or about the same?
It's a little bit better. When I can't see out of my right eye, I can barely see at all. So
I didn't know you got migraines.
I don't very often. I think it's just everything from the last couple of weeks catching up to me because I had like 5 games in 7 days, and then I didn't sleep very much for like 2 or 3 days because I had to study for everything that I was trying to make up. So
And then weather change, abrupt weather change is a big trigger for migraines in some cases.
it happened once last school year, and then
All right, well, take care of yourself and heal, and we can Zoom today, and that's what we'll do. Very good. Let's go ahead and have a word of prayer, and then we'll dive into this. Dear Father, Thank you for the glorious weather. Thank you for a glorious weekend. Now, Lord, help us finish photosynthesis and get into amino acids. We pray for the test on Wednesday, that it goes well for everyone, and thank you for this class. We pray for Jill's at work on Thursday, and for the rest of the semester, do watch over us. Help us stay healthy. In Jesus' name, amen.
By the way, the bacteria is the right thing, so today I'm gonna check to make sure that phenothiazine actually works again.
Praise the Lord. So, okay. All right. Photophosphorylation. So we talked about the OEC, the oxygen evolving complex. It is a very complex manganese oxygen active site in a relatively small protein. Protein is only 60, 65 kilodaltons, but it's the changes in the oxidation state of the manganese that drive the oxidation of water to molecular oxygen. And that oxidation of water to molecular oxygen will produce molecular oxygen that is a huge contributor to the ecology of the earth in general. The passage of electrons through electron transport chains, whether they be in mitochondria or in the thylakoid membranes, will be used to produce a proton gradient. And that proton gradient can be used to make ATP. If it's in the mitochondrion, we call it oxidative phosphorylation, because you're oxidizing reduced carbon compounds to generate the high energy electrons. In plants, the high energy electrons come from pigments that are found in the reaction center. The illumination by light, the reception of the exciton changes the reaction center pigment from a poor reducing agent to a good reducing agent. That electron is used to sent through the electron transport chain and used to produce a proton gradient. That is called photophosphorylation. Photophosphorylation. So the mechanics are pretty similar. Now you might remember maybe some of the things that you have to have in order for ATP synthesis to occur. And if we just do a quick review of those things, you'll see that they're all here again. So remember, one of those things is that you have to have electron carriers that alternate between those that carry just electrons and those that carry electrons and hydrogen ions. This alternation will allow you to dump the hydrogen ions on the other side of the membrane. Two, you need an intact biological membrane. Probably should have said this one first. An intact biological membrane with the electron carriers embedded in it. Three, the electron carriers must be arranged from those with a relatively negative, that's supposed to be a delta delta with a relatively negative biochemical redox potential, standard biochemical redox potential, to those with a relatively positive. This way, the transfer of electrons is thermodynamically, down the chain is thermodynamically spontaneous.
And these are the things that are required for photorespiration.
For oxidative phosphorylation, for photophosphorylation, if you have electron transport chains that'll pump protons, and then those protons will be used to make ATP, you must have these three things. So a thermodynamically spontaneous transfer of electrons down the, I'm getting tired of that jumping out on me, down the electron transport chain. Those and with, yeah, that comes from medicine. That comes from studying with med students and teaching med students and doing graduate work at a medical school and what have you. You pick up stuff and you don't. I know a lot of it. I don't know all of it. For example, you'll see this sometimes on a prescription. Know what that means? Twice a day. 3 times a day. 4 times a day. Daily.
Can you go back to the bottom?
Yeah. That's just stuff you pick up. Or NPO. Nil per os, that means no fluids or food before procedure, NPO. And it comes from a Latin phrase. If you ask most doctors or nurses what it stands for, they don't know. They just know, well, NPO means nothing, no food or water by mouth. So anyway, all right, so that's hang in there, Dane. All right, so if we get back then to this. Now in mitochondria, you might remember, the protons were pumped into the intermembrane space. In chloroplasts, you're going to pump the protons inside the lumen of the thylakoid. That's a much smaller space. So you're now going to pump protons into a small space, and you're going to have a whopping pH difference between the lumen of the thylakoid and the thylakoid stroma and the chloroplast stroma. So the protons get exuded to the opposite side of the membrane. That gives you proton motive force. You use that force to do work. So here essentially is the way you will transfer these protons. Oh goodness, what did I do? Stop, 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 stop. There. So we have PS2 here. We give our electrons to plastoquinones, a lot like ubiquinones. These carry both protons and electrons. So here are your protons. You're going to get those protons from the stroma. You give the electrons to the cytochrome B6F complex. Cytochromes just carry electrons. The protons are extruded into the thylakoid lumen. The electrons go to plasticyanine and then to PS1. The pumping of these protons with the evolution of oxygen, that's going to liberate even more protons. These protons then will build up inside the lumen. This is a so-called P side. P side. These then will be pumped from the lumen back into the stroma by a F-type ATPase, which structurally is virtually identical, at least structurally speaking, with the F-type ATPase in our mitochondria. When these get pumped out, you make ATP. The proton motive force in this case is even greater than it is in mitochondria because you're pumping the protons into a small sealed space. So the delta pH between the stroma and the lumen or the thylakoid is huge relative to mitochondria. So this process is called photophosphorylation, as we said, and the ATP synthase molecules in chloroplasts and mitochondria, structurally speaking at least, and mechanistically speaking, are essentially identical. The difference is where you pump the protons. In the proton, you're pumping it to the intermembrane space. The protons then get readmitted into the matrix. In chloroplasts, you're pumping protons into the thylakoid lumen, the P side, and then you're pumping them back into the stroma. And with bacteria, essentially, you're pumping protons into the intermembrane space between the peptidoglycan and the plasma membrane, and then you're pumping the protons back in using an F-type ATPase. Yeah, it is. It is. From your book. So the stoichiometry of photosynthesis has been extremely well worked out. This is due to the fact that you can isolate chloroplasts easily, and then from chloroplasts, you can isolate intact thylakoid membranes fairly easily. So because of that, the stoichiometry of proton to ATP ratio has been really, really well worked out. You get a very, very high hydrogen ion concentration inside the thylakoid lumen. The pH is essentially 3, whereas the pH inside the lumen, the stroma, is almost slightly basic. So again, you get this whopping pH difference, you get extensive ATP production, and essentially you get 3 ATP for every molecular oxygen you produce. That's more than you get per mitochondria. Non-cyclic electron flow. So the electron flow that makes ATP, that's cyclic. Cyclic photophosphorylation, cyclic electron flow. The non-cyclic flow will be used to make NADH. And NADH will be used for the fixation of carbon in the Calvin cycle, which occurs in the stroma of the chloroplast. So where you need the NADH, you make it. And it's also used for other biosynthetic purposes as well. Cyclic electron flow just makes ATP. And plants can switch between the two depending upon which one they need more. So in the cyclic form from PS1, those electrons go from the cytoferredoxin back to the cytochrome B6F and then back to the PS1 reaction center. And so this shows you essentially how this works. So you get a photon of light, you convert PS1 reaction site to a very good reducing agent. It reduces the two thiophytins, then it reduces the ferredoxins. The ferredoxins will then flow to a to an iron sulfur protein. From the iron sulfur protein, you go to a ferredoxin, back to the cytochrome B6 to the plastocyanin. Because electrons that come through the cytochrome B6 and the plastocyanin normally go to the P700 anyway, this detour simply gets you more hydrogen ions pumped into the thylakoid lumen for ATP synthesis. They don't make reductant, but notice you still need light to make this happen. Because that electron doesn't come for free. You have to change this from a poor reducing agent to a good one. You need the photon of light to do that. But light's free. And it's used to make ATP. So this is cyclic. Electron transport is just used for ATP synthesis. Yeah. These, so this is an iron sulfur protein here. Ferredoxin is an iron protein. And the ferredoxin oxidoreductase, that's an iron containing protein too. Yep, lots of iron. And then the chloroplast, of course, has magnesium at its center. Did you ever heard in 112, did Dr. Wyman tell you about C. Hopkins Cafe? Did, so. And let me try and remind you. So the elements that are required for plant growth are carbon and hydrogen and oxygen and phosphorus and potassium and iodine and nitrogen and sulfur. And then you also need calcium and iron and magnesium. And the way you remember this is C. Hopkins Cafe. Mighty good. Those are all the elements that you need essentially for proper plant life. C. Hopkins Cafe, mighty good. Very useful mnemonic. You've not heard that before. Okay, well, now you have.
I thought it was a person.
I thought it was too, or like a disease.
No, it sounds like Hopkins is a name. So now, when we look at bacteria, we notice that bacteria use hydrogen sulfide instead of water. They need something to re-reduce their reaction centers. And the hydrogen sulfide or elemental sulfur is used, and there are cyanobacteria that can use hydrogen sulfide under strict anaerobic conditions. That has been shown in several labs. Jack Meeks showed that at UC Davis when I was there. He forced these bacteria to grow with everything, except he gave them hydrogen sulfide, and it was clear they were making sulfate from it. Excuse me. Then he used radioactive sulfur, and he discovered that the radioactive sulfur that he gave them in the form of sulfide came out as radioactive sulfate. And that showed that these, also there was no oxygen being produced because the conditions under which he grew them, he had redox dyes that would grab any oxygen. And so no oxygen was being produced. The dyes didn't change any color. And they were making sulfate. And it was clear that the sulfate was being made by the photosystems. And then I told you about the bacteria in Solar Lake that also use hydrogen sulfide. And so it's possible that photosynthesis of this type was anoxygenic and then only became oxygenic later. And that eventually was responsible for the oxygenation of the atmosphere to provide the types of multicellular life that we have today.
So in general, oxygen is far more prevalent than sulfur, isn't it?
Yeah.
Can you say the two levels of the lake where it's getting those?
Oh, the hypolimnion and the epilimnion. And then the transition site in between, that's the site where the cyanobacteria grow in Solar Lake. And we know from essentially historic geochemistry, the rocks that you see in the earliest deposits of the earth were clearly deposited in very low oxygen tension, because you don't see the oxidized forms of iron that you see today. It's only when you start to see so-called red beds. And the red beds start to form about 2.5 billion years ago. That's clear that oxygen levels really started to rise, because the banded iron formations show that the oxygen levels at that time, which were the banded iron formations, the BIFs, they formed before the red beds. So banded iron formations don't form in oxygen levels we have today. So the red beds, then you start to see a little bit later, and then, you know, today start to see fully oxidized forms of iron that you don't see in the earliest fossil record. So by that indirect reasoning, it's thought that oxygen levels are very low in the early atmosphere and then started to become just gradually higher. And the big boost in oxygen seems to have been biological organisms, not any sort of chemistry. Yeah. All right, so there is a use of light that you see in the halobacteria. I don't know if any of you have ever flown over Salt Lake City in a plane. If you do, when the plane flies over the Great Salt Lake, the salt flats, you'll notice the salt flats have all these red spots on them. And those red spots are these halobacteria that grow in these very, very high salt concentrations. Most organisms can't grow in that kind of salt, but these organisms, these halobacteria, have to have high levels of of salt in order to grow. So they live in a very inhospitable environment. There's not a lot of food in these areas. So what do they do essentially to be able to grow? They have a way of supplementing their energy metabolism. And it's clear that oxygen is not very soluble under these very high salt conditions. Salt conditions of up to 4 molar. That's, yeah, crazy high. So they supplement their energy metabolism using this red pigment in the membrane called bacteriorhodopsin. Now, why is it called bacteriorhodopsin? It's called bacteriorhodopsin because structurally it has similarities to the opsin proteins in the retinas of our eyes. And these bacteriorhodopsins are light activated proton pumps. They take a photon of light, they pump a proton into the periplasm, and then that proton in the periplasm creates proton gradient that's used to make ATP. So the structure of this protein has been worked over ad infinitum, and as you can see, it's extremely rich in alpha helices. So you have helix 1 right here. Then you have a helix 2, which swings over here. Sorry, that's 1, that's 2. I'm getting these confused. That's 1, that's 2. Then this swings over here and around and over here. Here's a helix 3. That connects over here. There's a helix 4 behind there. That swings over here. 5, and then that connects over here to helix 6, like that. So this has several different alpha helices. And these alpha helices form a channel. And in this channel, you have these acidic and some basic amino acids. And there's a little retinol in the center here as well. And so the bacteriorhodopsin, in the absence of light, it's in the trans formation. The absorption of light causes a cis trans isomerization. And so the retinol goes from trans to the cis configuration. And that bacteriorhodopsin is attached to a lysine residue. If this sounds familiar, it should, because this is precisely what happens in our eyes. And again, this is taken from your book. So here's the retinol. And then you have a threonine and an aspartate and an arginine, and then these glutamates. Photon of light will cause this isomerization. That proton then is going to go to the aspartate, which then displaces a proton in the arginine, which displaces it to a tyrosine, which displaces it to the glutamate, and the proton gets released to the outside. And then a proton from the other side will come and protonate this here, because those free electrons are there. And then it shifts back again to this original conformation to get the absorbed photon of light, and the whole thing repeats. By playing essentially hot potato with these protons, one loses a proton, so one gives a proton to another, to another, to another, to another, to another, and a proton gets extruded across the membrane. You make maintenance energy for this organism so it can survive in this very inhospitable environment.
So these organisms use light but aren't photosynthetic.
No, because they don't have photosynthetic pigments. They don't have electron transport chains. They just use this as a low efficiency proton pump. And it's clear this supplements their metabolism. They don't live on this solely.
What else do they do?
They live on simple carbon compounds that are usually brought to the salt by the wind. And so that includes short chain fatty acids and amino acids and things like that. These don't grow terribly quickly, as you might guess. And that is photosynthesis. Any questions about photosynthesis? Dane? No? He's shaking his head. All right then. And now for something totally different. Okay, nitrogen. Nitrogen is going to be used to make amino acids and nucleic acids. Nitrogen is very abundant in our atmosphere, but atmospheric nitrogen is very stable. It's very inert. So in organic chemistry, if you use nitrogen to make a reaction go, what usually happens? Nothing. Nothing. Oxygen, you get combustion. Nitrogen, nothing. You dry things under nitrogen. It's so darn stable. So trying to get atmospheric nitrogen to react for biological purposes is really hard to do because it's so inert. But you need nitrogen. So you have to have proteins, which are made of amino acids. You have to have nucleic acids. They both contain nitrogen. So you have to get nitrogen somehow. So the acquisition and the metabolism of nitrogen and nitrogen containing compounds is a very, very huge part of metabolism. And then dietary amino acids, they get degraded to alpha keto acids. That ammonium will be ultimately released as ammonium. And to show you essentially biochemically how this happens, so from intracellular protein, you degrade those two amino acids. That degradation is a result essentially of proteases. Oh, this is hard to write with the mouse.
I'm sorry for laughing. It's kind of
Proteases. That's actually one of my better ones. It's hard to draw by the mouse. Those are degraded to amino acids. Dietary protein also is degraded to amino acids. Now, can those amino acids be used directly for protein synthesis? Of course they can. But the amount of protein you're going to take in a diet, let's say, for example, you're a cheetah. Right, meow. Your diet is going to consist of meat. You're a predator. So animal flesh consists largely of protein. Those amino acids can be used to make protein, but you'll have far more amino acids than you need for protein synthesis. Those other amino acids for metabolism, they'll be deaminated to ammonium. Ammonium is toxic to cells. It's toxic to cells. So if you are an animal and you excrete ammonium and you live in the water, no big deal. Because usually you have gills, you can excrete it straight out your gills. Or you could simply, because there's so much water, you could just excrete it directly in your urine. Because water is not a problem, because you've got so much water, you just take in more water, it's not a big deal. So if you are a aquatic critter producing ammonium, not a big deal. Because you got all this environment where you can excrete it, not a problem. So many fish, many amphibians, many aquatic invertebrates, they just excrete ammonia.
What about aquatic mammals?
Aquatic mammals don't. Aquatic mammals don't. Now, if you're terrestrial, you've got a problem. Because water loss is an issue for terrestrial creatures. You don't have water all around you. You have to be able to save water. You can't store ammonium either. It's too toxic. So what are you going to do? So you're going to convert it into a compound that is much less toxic and can to some extent be stored, called urea. And you're going to use the urea cycle to do that. Urea is nicely excreted. It's the major nitrogen product that's excreted by mammals, at least. And you'll make it by first creating this compound called carbamyl phosphate. The urea cycle is going to occur mainly in the liver. Other cells have the enzymes to execute the urea cycle, but the main processor of ammonium will be the liver.
Where's urea excreted from them?
Urine.
Okay, that's what I thought.
That's why when urine sits around for a while, then it starts to smell like ammonia.
Okay, that's
Because
what I-
urea breaks down because bacteria will break it down using the enzyme urease to break it down to ammonium and CO2. Now there's a shunt that's going to shunt carbon from the citric acid cycle to the urea cycle and back. And we're going to see some common compounds that are shared by these two. So your citric acid cycle is going to scratch the urea cycles back, and the urea cycle is going to scratch the citric acid cycles back. Where do you get the carbon from? The alpha keto acids generated by deamination of the amino acids are fed into the Krebs cycle. That creates CO2, water, and ATP, and then ultimately oxaloacetate that can be used to make glucose. And that's why some amino acids are gluconeogenic. Glucogenic, they used to make glucose. Not all of them are, but many of them are. Okay. Making urea is a strategy. It's the main strategy for terrestrial organisms, particularly mammals. It's not the only one. So as we said, if you are a monotelic, you excrete ammonia. As an ion, very soluble, not an issue. So your aquatic vertebrates, things like bony fish, the larvae of amphibia, and so on and so forth. They excrete ammonium, no big deal. Urea. Ureotelic animals, that includes a fair number of your terrestrial invertebrates, at least mammals, also sharks. Sharks concentrate urea in their tissues. That's one of the ways they regulate buoyancy, because sharks don't have a swim bladder. The way bony fish do, yeah. Now, uricotelic animals, that includes birds and reptiles, they make uric acid. Uric acid is not at all soluble. This is excreted, therefore, as crystals, and it uses very little water. This is one of the reasons why bird dung will etch the paint on your car, because of the acid in it, uric acid. Now, we make uric acid as well. We make it from the breakdown of purines. It's not our major nitrogenous waste, though. For reptiles and birds, it's the major nitrogenous waste.
Is that why when those kinds of animals and other things are dehydrated, that it almost comes out as a solid? Yeah.
Yeah, exactly right.
They get like clogged in the back. I know reptiles get clogged on the back of their legs.
Yeah. Yep. Exactly right. Uric acid is very poorly soluble. All right. Let's review just a bit about protein digestion. The protein digestion begins in the stomach. You don't have any protein digestion in the mouth or esophagus. In the stomach, you have stomach acid that is made by so-called parietal cells. Parietal cells. They secrete hydrochloric acid. The low pH of the stomach will kill bacteria, denatures proteins, and then allows the enzyme pepsin to degrade proteins. Pepsin is made by the chief cells. Chief cells. They secrete pepsin as a precursor called pepsinogen. Pepsinogen at low pHs auto-activates itself. It clips its own amino terminus off.
Pepsin is the ingredient of bismol, is it?
No,
No.
bismuth sulfate
Okay.
is the main ingredient of Pepto-Bismol. It's called Pepto-Bismol because it quiets the peptic acid. and helps with peptic ulcer disease and what have you. All right. Yeah. Pepsin degrades proteins into smaller polypeptides. Pepsin will cleave proteins on the amino terminal side of residues like phenylalanine, tyrosine, tryptophan. So you go from large protein chains to smaller polypeptide chains. Those acidic components of the stomach pass into the small intestine. And then a hormone, once those acidic components get here, a hormone called secretin is released. Secretin stimulates the production of bicarbonate by the exocrine cells of the pancreas. Secretin.
Is secretin listed on there at all?
No,
Could you write it down?
sure. Zoom really makes this computer go slowly.
Yeah. They could do a big thing.
Secretin. So the cells of the duodenum so-called enteroendocrine cells of the duodenum secrete secretin into the circulation. So this is an endocrine hormone, and it signals to the exocrine pancreas. Remember, the pancreas is a two-tone organ with an endocrine and an exocrine set of cells. And the exocrine cells will now release bicarbonate. And the bicarbonate is going to neutralize the stomach acid. Now, if there are amino acids in the duodenum, then these enteroendocrine cells will secrete another hormone called cholecystokinin. Cholecystokinin. Cholecystokinin will now stimulate the exocrine pancreas to release digestive enzymes. So secretin and cholecystokinin. Secretin and cholecystokinin. And I believe,
So, the cells secrete the, cholecystokinin.
right, enteroendocrine cells in the duodenum detect amino acids and secrete cholecystokinin. Doesn't take very many amino acids for them to do this. And cholecystokinin signals to the exocrine pancreas to release digestive enzymes. Cholecystokinin also signals to the gallbladder to release bile in the presence of fat. So that's another thing it does. This thing serves a couple of purposes. And cholecystokinin is often abbreviated CCK because cholecystokinin is a bit of a mouthful. All right. So if we go back to the slides, we can see that the pancreas, the exocrine pancreas, secretes these digestive enzymes into a ductal system. That ductal system will then feed into the duodenum, and these are released as inactive precursors called zymogens. Zymogens. The pancreas does that to prevent the pancreas from digesting itself because these enzymes are very powerful. So it releases these enzymes in a zymogen form. They need to be proteolytically processed in order to become active. And the way they become activated is, frankly, rather interesting. All right, let me first talk about some of what goes on in the stomach. So in the stomach, if you look at a slice of stomach, you'll notice that it's pitted like this. And then if you take a section through those pits, you'll notice that these pits actually are a column of tissue. You have different cells like this. They go down and then weave about like this, and then make another column. And up here, this hole here is those pits. These pits are called gastric pits. Gastric pits. Now, these pits are lined with cells, and these are all different cells. So one of the group of cells that you see are these cells that are filled with kind of a mucousy, and the cells are shaped like a champagne glass. That's why they're called goblet cells. Goblet cells.
The mucus cells are also called goblet cells.
Goblet cells, yeah. Applause, please. Goblet cells make mucus. Mucus is hugely important for the life of the stomach because that mucus is going to coat this surface and prevent it from being damaged by the acid. You also have these parietal cells that make the acid. Notice a parietal cell has got kind of a fat look to it. That, in fact, is one of the ways you tell a parietal cell. Tends to be kind of fat looking. So these are these two here, next to each other, both parietal cells here. There's one. Those are all parietal cells. Parietal cells make acid. They make acid. And then the chief cell, as we said, the chief cell makes the pepsinogen. Pepsinogen. Chief cells are pretty, chief cells are full of granules. That's one of the ways you may identify a chief cell. They secrete pepsinogen. Pepsinogen. So these are full of granules here. Those are probably chief cells. That's
Can you
probably one there.
spell
Pepsinogen again.
Dan, you look like you're ready to fall asleep.
I'm awake. Just
Yeah, headaches are no fun.
yeah.
Pepsinogen. And pepsinogen is processed to pepsin. Typically, if you see O-gen, that means it's a zymogen. See O-gen? So that O-gen ending usually means it's a zymogen. So for example, the pancreas secretes trypsin, and it secretes it as trypsinogen. It secretes chymotrypsin as, guess the name? Chymotrypsinogen. And so on and so forth. So O-gen means it's a zymogen. All right, and there are other ways these cells are signaled by the vagus nerve and by other gastrin, which is made by G cells and blah, blah, blah. I don't want to belabor that point here. Instead, I'll belabor it here. So with the cells of the stomach, you have innervation of the so-called ECL cells. These are enterochromaffin-like cells. That's why we call them ECL cells. ECL cells make histamine. Histamine. Now, histamine isn't just for achoo, achoo, achoo. Histamine is also a neurotransmitter, the signaler. And histamine is going to signal to the parietal cells to make acid. The vagus nerve is also going to innervate G cells. They're going to make an endocrine signaler, a small peptide called gastrin. Gastrin is an interstomach signaler, an intergastric signal. Signals to the ECL cells to make histamine. Histamine causes parietal cells to make acid. If the acid causes the pH of the stomach to get too low, it shuts down. Feedback inhibits, negatively feedback inhibits the production of gastrin. Whereas amino acids will stimulate the production of gastrin. So, have you guys heard of a drug called Zantac or Tagamet? Those tasteless commercials on TV with the taxi driver who had the pastrami on rye and now has the upset stomach and gets the advice from his charge. Here, take this. No, I should see a doctor. And the guy says, you just did. And he gives him Zantac. Zantac is rimantadine. Rimantadine inhibits the H2 histamine receptor. The H2 histamine receptor. Loratadine, fexofenadine, cetirizine, Benadryl, things like that. they inhibit the H1 histamine receptor, completely different receptor. That's why Tagamet will not help your allergies. That's why Benadryl will not help your GI tract. Because they bind to two completely different receptors.
I'm curious, so there's gastrin,
Gastrinogen, no. Gastrin is made as an active signal. Some people develop so-called gastrinomas. These are tumors of the G cells. And those gastrinomas cause excessive acid production that cause peptic ulcers.
So is gastrin upregulated or downregulated in CL?
Upregulates.
Okay, so when the systemic pGH is too low, then
Then it inhibits production of gastrin, yeah.
okay.
So pepsin auto activates itself in the presence of acid. As we said before, remember it's made by the chief cells. And there's a bit of physiology on here, but essentially this just reiterates what we just said. Eating has these three phases, cephalic, gastric phases. During the cephalic phase, you see food, you smell food, you start to get hungry. You see a commercial for Dairy Queen or something like that, or what have you. Or you walk by the DC in the morning and you smell pancakes or something like that. Or what have you. That's the cephalic phase. Even when the food actually gets in your mouth, you taste it, you start to salivate, that's the cephalic phase as well. Once the food hits the stomach, that's the gastric phase. So the chyme that enters the duodenum causes those enteroendocrine cells to release secretin, and then amino acids or fat will cause the production of cholecystokinin. These enter the bloodstream, they signal to the pancreas, and in the case of cholecystokinin, also the gallbladder, and you get the production of secretin will induce bicarbonate, cholecystokinin will induce the enzymes that'll break down fats, proteins, things like that. And this simply shows you how those zymogens are activated. There's an enzyme on the surfaces of the intestinal endothelial cells, intestinal enterocyte cells. This enzyme is called heterokinase. It's membrane bound. It's a brush border enzyme. Heterokinase activates trypsin by clipping the N-terminus off trypsinogen, turning it into active trypsin. Trypsin will then activate all the other zymogens. So it does this in a stepwise fashion. The other enzymes only get activated if trypsin is activated. Trypsin is only activated when it hits the small intestine by heterokinase. And now you have amino acids. What do you do with them? And that's going to be the subject of. Because we're out of time.
I have a question for you. I saw this thing on the internet. I don't know if it's true or not. Somebody suggested not eating berries and oatmeal at the same time because they said that like the acid from the berries prevents breakdown of the oatmeal in your mouth. And you like break down the oatmeal less and it takes you longer to digest it.
The amount of starch digestion in your mouth
So
is not that great compared to the amount of starch digestion that occurs in the GI tract.
okay.
Secondly, yes, your mouth pH tends to be more alkaline, but mouth pHs differ. And also the acid in berries is not that great.
Okay.
So I would label that as baloney.
Thank you. I saw it and I was like, that just doesn't sound right, but like maybe.
Did you hear that question, Dane?
Eating oatmeal and berries at the same time
Yeah.
causes what?
Yeah, that's it prevents you from properly digesting the starch in the oatmeal.
Oh,
That's balderdash,
yeah.
pure balderdash. So eat your oatmeal and your strawberries and your blueberries and be healthy. And have no guilt.